Next speaker is Parag Katira.
Perfect. Really excited to be talking here, and thank you for organizing this. So I'm Parag Katira. I'm from San Diego State University, and hey, there you go. So mostly what we do in my lab is look at the mechanics of cancer. So we talked about a lot of things in terms of what's going on in the biology and how do you model it. We look at it from the physics and the mechanics side, all the way from what is happening at the cellular level, right? So all the signaling, biochemistry, how that affects the elements that generate the forces and give the cell its physical characteristics. The cytoskeletal elements, how they organize because of whatever is happening in the biochemistry, how the cells generate forces, how it interacts with the environment because of these. Then looking at single cell level force generation, cell migration on substrates with different stiffnesses. We can also take it to 3D. So you have a bunch of red cells that are chemotaxing towards a chemical signal being released at the gray plate. As they move around, they're moving in a three-dimensional matrix with fibers everywhere. And they change this matrix as they move. And what they're doing is that it's sort of pushing the blue cells away from wherever the chemical signal was coming from. We can also do collective cell migration. So you have a cluster of cells migrating. They can migrate in different ways. You have one leader versus multiple leaders that nudge things around. Again, in a three-dimensional fibrous matrix, so you can change a lot of things about the matrix, the fiber orientation, fiber density, cross-link density. You can look at what happens in tissues collectively where cells are interacting with each other. Again, focus is purely on mechanics. So you have a group of soft cells, which is green, group of blue cells, which is blue, sort of competing against each other. All interactions mainly driven by the mechanics. And then you can also take it to 3D and look at tumor growth. You can see different morphologies evolve just by changing the mechanics of the cells. Focus of our lab is on looking at the mechanics of the cells. But I'm not going to talk about that. I'm going to talk about what we did in the IMO workshop in November, because that was something very new and unique for me. And that was the first time we was doing something at the organ level. Glioblastoma, I had no clue about glioblastoma, but this was a very good setup where I learned quite a bit. And we modeled glioblastoma as a function of the resource distribution within the brain, as a function of the stiffness of the brain tissue, and cells with different ploidies, basically, as Vural talked about it. He was on the team. So it was an awesome conference, except that we didn't have any immune cells. So no immune cells, sorry. All right. Every model is specific to answer a particular question. Otherwise, there's no value to it. And the question here was, again, driven by this polyploidy that you see in tumor cells. And the idea is that having genome doubling, right, having more nucleotides, having higher ploidy, basically gives them a bunch of superpowers that lets them escape therapy, treatments, or do something that is different and sort of survive more. It also makes them require more resources and changes the behavior under different environmental conditions. And the evolution of ploidy, again, is a function of what is there available in resources. And so the idea was to try and figure out what is this interplay between cell ploidy and the environment that they are in, and how does that sort of drive glioblastoma growth and progression, and what could it mean? So how that whole project started, so this was basically day one. We're trying to put the pieces together, and there was a lot of beautiful in vitro stuff where you had cells growing up in a chamber, able to migrate through to another chamber, and from there you could calculate or figure out sort of the proliferation rate, the migration rate as a function of ploidy and as a function of availability of resources. There was also clinical data where you actually had like patient scans, which was very scary for me. I've never worked with patient data before, and it's depressing, at least for me, but it's important. So it was, again, a good example. But yeah, so you had clinical data where you had actually patient level, MRI scans, PET scans. You could actually look at, there was MRE data available. So elastography, you could actually measure tissue stiffness. You could measure the amount of resources through PET scans, whatever. And then you had beautiful math models looking at single cell level dynamics. So what is actually happening in the in vitro data with the growth and the migration. And what we were trying to figure out was the ideal sort of the tumor level model. And again, there are a lot of beautiful tumor level models out there for glioblastoma growth in the whole brain. The fundamentals are pretty well established. You have these PDEs and ODEs that drive tumor growth, migration, diffusion. You can interact, sort of put in resources and all of that stuff. What we did was a little bit different in terms of just the model implementation and how we sort of simplify and different rules we put in. And what we came up with was this stochastic state space model. We call it stochastic state space model because we couldn't figure out if it was agent based or if it was continuous, something in between. It's something weird, but it works, I think. The idea is, so the elements of the model are, you have your cell populations. You can have multiple different ploidies. Then you have your dead cells. They all go together. Doesn't matter once you're dead, how many ploidies you have, how many genome doublings you had or whatever. You have your resources. You can get vasculature. Again, your brain has vasculature, but you can get extra vasculature, which is being recruited by the tumor. That's what we focus on. You have the tissue stiffness. And as the cells move around and distribute, they actually alter this environment. And then you have obviously treatment. And what you do is you take your brain tissue and you break it up into pixels, obviously not into this giant pixels, but about small, tiny pixels of 2 millimeters by 2 millimeters. It's a, if you can ask me why that was the choice, and I will explain it to you. But you break it up into these voxels or pieces. And then you define each voxel by the state or voxel state by the elements or the value for these different elements that I talked about. So different cells, the oxygen, the glucose, and phosphate. And then you update the state of each voxel, which is you update the state of every element in there using those equations. I'm not going to go through those equations. Again, we can talk about it more in detail if you want. But the key features are that each voxel is treated as a well-mixed system. So inside the voxel, everything is mixed. Cell death inhibition is based on resource availability in the voxel and the carrying capacity, where carrying capacity is specifically just the maximum amount of cells you can pack in, just a spatial carrying capacity. The other part is coming from the resources. Okay. And then your cells can actually convert. So if you starve them of resources, you have a higher likelihood of them gaining ploidy. If they're stressed, you have stochastic migration of cells from one voxel to the other based on, again, resource availability. So this is a sort of a biphasic, we'll talk about that, sorry. And the next slide. And then also based on the stiffness of the environment that they're going to or they are in, what they prefer. And then there is stochastic recruitment of vasculature, depending on, again, what the cells are, how stressed they are, and what is going on with the treatment and the therapy that you're giving. So you could, if you're sort of giving radiation therapy, or then that would kill the vasculature. If you're doing surgery, then that would take away all vasculature, all cells, and reduce the stiffness. So you can put in those different conditions. Oh, I had these beautiful highlights, but anyway. All right, so to start off, we had this brilliant collaborator. So Frederica, she went in and got from wherever MRE images as well as PET scans for young, old, male, female. You could sort of stratify them and average them to get the stiffness map and the average resource distribution map for whatever age, gender, and however you want to do it. And then this is just a representation. So you can start off in any voxel about close to somewhere around 15 to 20,000 cells in there. And you would see this tumor sort of grow and spread around and do whatever it does, right? The cool part about this, about the implementation, was that you could simulate 365 days of tumor growth in less than 5 seconds. You hit run and take a yawn and it's done. So that was pretty cool and useful. And obviously, the model is complex. There are a lot of things we added just for fun because we could. So a lot of parameters, and we'll talk about the parameters in a bit. If you add more ploidy and more cell populations, then you obviously can add more parameters. But anyway, so some basic assumptions we made initially was that if you have higher ploidy, that means you need more resources. Now, how cell migration works with resources was based on this go versus grow. So if you have enough resources in the environment, then you don't care about migrating anywhere else. You're happy there and you're going to divide and you're going to do whatever you want. It's when you are at this, if you're obviously at very low resources, then you don't have the resources to migrate and you're not going to migrate. And you have this intermediate value of resources where it's like, it's enough for you to migrate and go to another place where you could find more resources. And so you have this biphasic distribution for migration speed versus resources, but higher the ploidy, you would sort of feel that stress early on because you need more resources. Okay. That was one assumption. The other one was that if you are under hypoxic stress, then you can increase your ploidy, but the more ploidy you accumulate, it's harder to increase your ploidy number. And the third one was that, yeah, having higher ploidy increases your resistance to chemotherapy. And so based on that, we did a simple test. So here we have 600, 2 years of simulation. At 300 days, you apply radiation therapy. So the tumor sort of dies down, a lot of cells go away, and then it comes back. And you can see that sort of towards the end, your higher ploidy cells are sort of starting to take over and going faster. All right, so this is, again, basic, just what can we do with the model? The other thing we could do is you could start off with just 2N population, see them at, say, 10,000 cells, put them in one particular walk cell, and see how your ploidy evolves over time. And you can do this for high resource brain or low resource brain, so young versus old or whatever. And you can see that low resources sort of pushes higher ploidy over time. And you can see where the cells are different ploidies, different cells are sort of distributed around the brain tissue and brain region. And the last thing, again, we did was you take a young brain tissue and old brain tissue, differences based on stiffness and resource availability. You have more glucose in the old brain. Start off the tumor at the same point, and what you see is then that in the older brain tissue, you end up with higher ploidy cells at a larger number. Again, consistent, you can run 20 simulations and average them out, and you'll consistently see this happening. So this was day 4 of the workshop. And that's where we left. But then we continued working on it. And it was sort of trying to figure out what should we do with the model, right? And trying to get it to have some value and be more applicable. And so this was actually clinical data where you have a patient, you have the ploidy measure, the patient comes in, you have your first resection, and then you have 415 days at which the patient comes in again, the tumor has come back. There is treatment in between, and you have the second resection. And so step 1, get all the parameters. Could find a lot of at least broad estimates for the parameters in the literature, but there were quite a few other ones we couldn't and we guessed. We also got some from the in vitro data. And then the idea was, okay, so you take your input, which is after the first surgery, you have your cell distribution. You take that in, you plug it in, and then you see, so this is actually post we'll get there. So that's treatment that's happening around starting day 37. Cells are gone, dead, and then you'll see that they will start coming back and grow. And the idea is that whatever you end up with at day 415 should sort of match what you see in the sort of towards the end, right? And you have some variable parameters. The idea is that you fit the parameters that you don't know. And the way you fit it is you have what you see initially, what you see at day 415, and you're trying to recreate both the ploidy number distribution and also the cell densities within the map. And we were able to get pretty close. Again, this is the sort of the ongoing work, which we're very excited about. We are also doing some basic fundamental testing of, does the model do what it's supposed to do? For example, when you say birth and death and resource utilization, well, when you have enough resources, the cells are happy, and then the cells sort of start dying down as your resources get consumed. Also, there is diffusive movement, which works nicely. And then, yeah, we're working on further collaborating stuff and getting the results. I want to thank the entire team, Violet from the IMO. And then obviously that's my research group who keep the things running, because when I'm going and doing all these weird things, then they have to work on the actual funded projects. So thank you.
Well, I have a question.
Go for it.
So I have tons of questions, but let's just focus on the key question here. For the GBM model, you painstakingly at the very beginning, you had an MRE data for males and females and young and old. You had cross spectrum of ages.
Yeah.
Did you look at that, investigate those parameters and how that changed and their impact on the outcomes yet? And if not, sex differences, age matters.
I know. So we didn't do the sex difference. We did the young and old,
Yeah.
and that does have a difference. You start off with the exact same location, same initial seed, and you get different outcomes. Sex differences, we haven't. And sitting here, I was like, oh, we should definitely try that.
Yep.
We have the data. So yeah.
Yeah, that's
absolutely.
awesome.
Yeah.
It's great.
Thank you.